Okay, everybody, we get to some really good stuff today. It gets a little bit better than today, but today, this is good stuff. I want to talk with you today about DNA and how DNA turns out to be the thing that's carrying instructions from one generation to the next. All right, so that's the main menu item, and that's what this lecture outline is about. Before I get to that, though, let me go back to some things that I said that I would finish up with you about in lecture on Tuesday. I said, gee, let me tell you about this on Thursday. Let me do some cleanup work for what I was talking about on Tuesday. And I'm really just focusing here on the chromosomal alterations and the extra chromosomal inheritance. And then we'll talk about DNA. So I flashed this picture up at you. Chromosomal alterations. One of the kinds of chromosomal alterations that we see in humans is this idea of aneuploidy. Aneuploidy means that there's a non-usual or non-normal number of a particular chromosome. So Down syndrome is aneuploidy trisomy of chromosome 21. So that's an example of aneuploidy. And if you look at this picture here, you can see, in fact, that from this individual's cells, there's a pair of every chromosome except for chromosome 21, where there's actually 3 copies of chromosome 21. And so that in turn gives rise to some gene dosage problems. I talked with you about bar bodies a little bit on Tuesday. We can handle that problem, the gene dosage problem that women have, because they've got 2 X chromosomes. But there's nothing set up to handle that equivalent problem for having 3 copies of chromosome 21. All right, so there's aneuploidy. There are other types of alterations of chromosomes that cytogeneticists can recognize fairly easily by looking at those chromosome spreads, like the one that you saw on the previous slide. Looking at differences in the banding patterns. And here's a pretty exhaustive list of the types of issues that a cytogeneticist might be able to find. Again, look back at this, you can see that they're lit up with some different colors and some banding patterns here, red, green, red. A cytogeneticist might look at a karyotype. Incidentally, that's what this is, a photograph or an image of all of an individual's chromosomes, whether it's a human or a fruit fly. That's called a karyotype. When a cytogeneticist looks at these karyotypes, they might notice that some of these bands, the chromosome number is fine, but the bands aren't quite right. So you might notice a deletion. You might notice a duplication, right? Obviously, they're bigger chromosome, but you'll see the banding patterns repeat. Here, a smaller chromosome, and one of the bands is missing. Sometimes you'll discover inversions, and sometimes you'll discover what are called translocations, where a piece of one chromosome, chromosome 6 for instance, ends up being swapped with a piece of another chromosome, chromosome 16. Right? So a little bit that's usually on chromosome 16 now resides on chromosome 6, and vice versa. Right? So these are all different kinds of chromosomal alterations that cytogeneticists can find. Some of these are more likely to cause problems to an individual than others. Deletions almost always have some consequence associated with them. Duplications sometimes have complications associated with them, depending on the genes that are involved in the duplication. Inversions rarely give rise to problems for an individual. Their gene dosage is fine and the regulation of those genes is fine, but they often find that they have fertility problems. And so if you know a couple that's having a hard time conceiving a child, one of the things that will be looked at some point in the process is a cytogeneticist will look at the karyotypes of the individuals to see if maybe the woman's got some alteration with the structure of her chromosomes so that they don't match up well with the guy's chromosomes. Right? So something that can happen there usually doesn't have any consequences for the health of the individual, but can cause fertility problems. And the same would be true for those reciprocal translocations. Right? Gene dosage doesn't get affected, gene expression probably doesn't get affected, but it can cause problems during prophase 1 of meiosis, and also during mitosis when you're lining up these chromosomes and getting ready to replicate nuclei. So, chromosomal alterations, that's sort of the whole suite of the ones that you might see. And then I wanted to say also just a few words to you about this idea of extra chromosomal inheritance. You're all familiar with the idea behind Jurassic Park, right? That you get a blood-sucking insect trapped in amber, you extract DNA from the blood of the dinosaurs that the insect used as a meal, you take that DNA and you put it into some other thing, like what's the thing of choice, a chicken egg, an alligator egg, some reptile, typically egg, and you let that thing grow up and it becomes a Tyrannosaurus Rex as well as a number of other different things. Neat idea, probably not going to work in practice. And the reason it wouldn't work in practice is that we know a lot about the information that's stored in DNA. We're really good at figuring out what's encoded within DNA. We've got all sorts of methodology, all sorts of tools, lots of computer approaches to deciphering the information content in DNA. We got the information in DNA down pretty well. And so we focus on that. But we know there's a lot of other information that's passed from one generation to another that doesn't reside just in the DNA molecules. So the structure of the DNA molecule contains some information. In a little bit, in another lecture or two, I'm going to talk with you about the interaction of proteins with DNA in our chromosomes. That also has some information content associated with it. There's this phenomenon known as imprinting. We'll talk more about it when I get into gene expression discussions. But this idea of imprinting is kind of cool. Turns out about somewhere between 1% and 5% of human genes are imprinted. And what that means is this, is that the chromatin structure, the way the DNA gets packaged, is different in male germline cells relative to female germline cells. And those differences get passed on so that I got a copy of a gene, an imprinted gene from my mom, I got a copy of an imprinted gene from my dad. For my whole life, I preserve the chromatin packaging status that I got from my mom and I got from my dad, and they're different, and those differences can cause differences in expression. For imprinted genes, what that means is that I'll express either just my mom's version of the gene or just my dad's version of the gene, right? Same will be true for you. We've got 35,000 genes, 1 to 5%. We're talking here about hundreds of our genes we express in a way that's determined ultimately upon which parent gave us that copy of the gene, right? And so that's something you wouldn't be able to capture just by doing the Michael Crichton trick of taking the complete sequence of an organism and plugging it into some sort of fertilized egg cell. And there's other things that we know are associated that pass information from one generation to another. So there's a chromosome in mitochondria, right? That's not part of the nuclear genome, that's extra chromosomal. In some cases, there's a chromosome in chloroplasts, that's extra chromosomal. There's actually nothing at the level of DNA that dictates, that spells out how it is that you make a phospholipid bilayer. You know how you make a phospholipid bilayer, the cell's membrane? You add on to an existing cell membrane. So there's lots of structural information associated with cells that is not encoded with DNA. And so while we're really good, and it's easy for human scientists today, right now, to focus our attention on the information stored in DNA, I want to make sure you appreciate that there's more stuff going on than that. And we haven't really explored that other kind of information nearly as diligently as we have the information that's stored in DNA. So that's what's up with this idea of extra chromosomal inheritance. And now we can start talking about the stuff we really do know a lot about. And I want to take you by the hand over the course of the next 20 minutes or so and give you a sense of the history of how this rolled out. You know, the names of the researchers, it's not particularly important. I'm not going to really quiz you on their names, and the dates aren't particularly important. You don't have to know the exact dates, and I'm going to be checking my notes myself to make sure that I'm not telling you any lies as I go through this. So the dates and the names aren't especially important, but what I hope you take away from what I'll tell you in the next 15, 20 minutes is a couple things. The standing on the shoulders of giants sort of thing, right? That we have these incremental insights that help us learn and build from what, you know, in an incremental fashion. It's not like there was a bolt of lightning and Watson and Crick got the structure of DNA and everything was clear. There was a really incremental process by which things got built up to the point where they were poised, they were at the right place in time to be able to pull things together and come up with the double helix model, right? So I want to give you a sense of how it is that that progression happened. I hope you get a sense of how slowly this played out, the process of figuring out that DNA was such an important molecule. It played out over the span of about 50 years. All right? And so it was a slow process. And what we've learned in the last 50 years, since we've had that insight that Watson and Crick got about the structure of DNA, it's like 1,000 times more stuff has been discovered and learned in the last 50 years than in the 50 years that preceded it. So we're really at a stage right now where there's an exponential increase in our understanding and our appreciation of things. And I hope you get a sense of how much more quick the pace of discovery is now relative to what it was just 50 to 100 years ago. So again, I want you to have that sense. And then also there's some human drama associated with this, and so it makes for a good story. But let's get started. And the bottom line is simply this. And I'll put a little, I need to take a deep breath here, because that sentence, DNA is the genetic material, is a big stinking deal, right? When I say it boggles my mind that when I say that, I still get a chill up and down my spine. It's something that I have to restrain myself from doing because it's such a powerful statement. You guys may not appreciate it yet, but oh boy, people for thousands of years have been trying to figure out what it is within ourselves that encodes and passes on information from one generation to the next. We're really privileged to be living at a time, just the last 50 years or so, where that has been understood, where we know it is DNA that spells out those instructions, right? That's a big deal. Plato, Aristotle, lots of people have wrestled with it. And I've already told you that, in Plato's time, the prevailing thought, and this played all the way through to the early 1900s, so for a couple thousand years, the prevailing thought about how it is that you passed information from one generation to the next was something I think is ridiculous given what we know today, but the prevailing thought was that in every sperm cell there was a little tiny miniature version of myself, and in every egg cell there was a little tiny miniature version of some woman, and what would happen is those little tiny, sometimes animated and conversational, tiny versions of ourselves would blend together, sort of average together when they came together in a fertilized egg. I mean, that's how ludicrous the proposition was. That's the level of understanding that people had for thousands of years, and we're so much better off now that we know what's really going on. There's literally instructions that are spelled out in a molecule, DNA, that carries that information and passes it on very faithfully from one generation to another. So this idea that DNA is the genetic material, if you could have delivered that insight to Plato in some meaningful way thousands of years ago, where we would be today, it's hard to imagine, given how quickly we've been able to crack open lots of difficult problems once we've had that insight. So neat stuff, and we're lucky to be living at a time where we can be seeing the answers to these long-standing questions. To some extent, the process of figuring out that DNA was that key molecule began in the early 1900s with the work of a British physician, an army surgeon, whose name was Frederick Griffith. Griffith, this is at the end of World War I, right? Griffith noticed that a lot of his patients were dying from pneumonia. And he was able to determine a specific strain of pneumonia was what it was that was infecting these troops, Streptococcus pneumoniae, right? So that's a specific kind of pneumonia that his patients were suffering from, and many of them were dying from it. And he also found out that he could infect mice with the same bacteria, the same Streptococcus pneumoniae, that were killing these troops. Right? So he could have these mice and he could get them sick, and they would have essentially the same symptoms that the human patients would have, pneumonia, right? Difficulty breathing, lots of coughing, that sort of thing. And some of the mice lived and some of the mice died, just like some of the people lived and some of the people died who got infected with Streptococcus pneumonia. And he realized in the course of infecting these mice and playing around with them that when he would culture the bacteria, there were two different kinds of bacterial colonies that would grow up on his petri dishes. It was as simple as that. He needed to have fresh stocks of bacteria, so he would take an infected mouse, squeeze its lungs out after it had died, or sometimes before it had died if it wasn't going to die from the disease. But he'd squeeze out the juice from the lungs, and he'd put it on a petri dish. He'd get an individual colony, good standard microbiology practice. And when he would look at these petri dishes, he saw two different kinds of colonies. Sometimes the colonies of bacteria were smooth, and sometimes they were rough, right? Noticeably smooth, noticeably rough, okay? And he got him thinking, what's up with that? And, oh, here's a picture of it in printing. And then Griffith did something that I don't think would have occurred to me to do, right? I don't know, maybe he had a lot of time on his hands. I'm not sure what it was, but he became curious as to what was more dangerous, what was more devastating to the mice. If he infected them with the smooth bacteria, was the outcome different than if he infected them with the rough bacteria? Okay? And so he'd do both, and what he found time and again is that if he infected a mouse with the smooth bacteria, they died. Invariably, they died. And when he infected a mouse with the rough bacteria, they got sick, but they didn't die. Right? So he's thinking, oh, this is cool. Is there something we could do to make sure that the guys who get this disease only get it from the rough guys, the rough bacteria, as opposed to the smooth? He's starting to think that. And so that's pretty straightforward and pretty obvious stuff. I think we might all understand how it is that he would be interested in that. But then he does something that's really weird. He took heat-killed rough bacteria and squirted them into mice, and the mice didn't even get sick. And he took smooth bacteria, killed them with heat, put them in boiling water for a while, put that into the mice, and they didn't get sick. And so he's thinking, oh, I'm onto something here. If I heat these things up, if I bring it to boiling, I kill the bacteria, and the bacteria don't cause any harm. That's cool. And then here's the real strange leap. For some reason, he took dead, smooth bacteria, so he heat-killed them, put them in boiling water, and he mixed them with live, rough bacteria, and he took that mixture and squirted that into his mice. And guess what he found? I mean, so that, what's he thinking? What was the logical connection that made him mix dead, smooth bacteria with live, rough bacteria? I mean, coming into this, you got to be thinking, okay, these mice are going to get sick, and they'll get better because it's the rough bacteria that's infecting them. But that's not what he found. Strangely enough, why you even put this to the test is beyond me. I wouldn't have thought to do this. But he puts this to the test. He takes dead, smooth bacteria, mixes them with live, rough bacteria, squirts them into mice, and the mice not only get sick and they die. Where'd that come from? How'd that happen? And further, when he squeezes the pus out of the dead mice, the only kind of bacteria that grow on his petri dishes now are smooth. He only put live rough ones in. All the smooth ones were dead. How did he get live smooth ones out of these infected mice that had just died? What happened? How did the information that allowed the bacteria to become smooth get transmitted from a dead bacteria to the live bacteria, the live rough bacteria? That was a real puzzle. And when he did it the other way around, you can predict what's going to happen pretty easily, right? If you took a dead rough and you mixed it with a live smooth, you know what happens? You infect a mouse with that, they all die. And when you squeeze the pus out of their lungs, they've all got smooth bacteria, right? No big news there. The puzzle, the head scratcher, was he took dead smooth bacteria, mixed them with live rough bacteria, infected mice with that mixture. The mice died, and all that he got out was smooth bacteria. He coined a word to describe what he had observed, right? The word that he used was transformation. It's a British guy, it's an English word, makes sense, right? That's a fair word to describe what's happened here. Somehow or other, the dead smooth bacteria were able to transform the live rough bacteria into smooth bacteria. They were transformed. And they must have been transformed because some information, some heritable information, got transferred from the dead smooth bacteria to the live rough bacteria. The transformation must have been a genetic event. It is a heritable transformation, because from that point forward, those smooth bacteria stayed smooth. They were able to faithfully pass that on to subsequent generations. And Griffith then says, check this out, folks. And by the way, if you're looking at these pictures, this is a cleaned up version of the picture. In earlier versions of your textbook, I used to love, this was one of my highlights of the year for me, they would have like an X on the eyes for the mice to let you know that they were dead. It was so cool. It was so like 1940s kind of way to convey to you that the organism was dead as opposed to sleeping. So I hope that you can envisage what a little black X would look like there, because those are dead mice in these particular pictures when they're curled up that way. Small things amuse me, right, I suppose. So what Griffith takes away from this is he coins this word transformation, and for the first time he suggests that maybe the thing that's carrying information from one generation to the next, and the thing that's carrying information from the dead smooth bacteria to the live rough bacteria, maybe it's DNA. And the reason he says it might be DNA, as opposed to the alternatives, things like proteins and sugars, which people had been considering pretty actively at the time, is that those kinds of molecules, proteins and oligosaccharides, sugars and lipids, those were known to be damaged by heat, right? DNA was known to be fairly resistant to problems that might come from being exposed to heat. And so of the four molecules that people thought of as being abundant within living cells, only one of them seemed to be able to withstand exposure to heat, and that one was DNA. And so for the first time, Griffith says, gee, I know a lot of people are putting their money on proteins, because proteins look an awful lot more like they have the capacity to store information than DNA does. And I know some people are putting their money on sugars, and some people are putting it on lipids. It looks to me like those three guys aren't much in the running, because they all get damaged by heat. DNA doesn't. Maybe it's DNA. Okay, so that's the first glimmer of an insight in the mid-1920s, that maybe the action for heredity and for storing information, maybe it's DNA. And so I said a lot of people at the time were thinking proteins would be a better bet than DNA. You know why proteins looked more promising, and they probably still look more promising in some sense today? There are 20 different amino acids, okay? And so when you want to, there are 20 different amino acids. There are only 4 nucleotides, G, A, T, and C, guanine, adenine, thymine, and cytosine, right? Think about our alphabet. I'm going to take a guess here, but I'm put myself out there? 26 letters? Good, right? Okay, good. People are nodding. You think that I don't know. You think that I'm joking. I don't know. I have to figure this out every time. And if you want, I can, take off my shoes and socks, and we can A, B, C, D, E, F, G, and we can count it. But so 26 letters. You can do some neat stuff with an alphabet that's got 26 letters. What would the consequences be if your alphabet was just 4 letters, right? If I put you on a desert island and I say, okay, now here's the deal. You get off the island when you can start to convey information with a 4-letter alphabet instead of a 26-letter alphabet, right? At first, you think, okay, this is stupid, what a ridiculous artificial constraint this is, but if I'm serious and you're not getting off the island until you can convey some specific information with a 4-letter alphabet, you know what you do? You eventually roll up your sleeves and you start spelling things out with your 4-letter alphabet, but your words become really long, right? That's the consequence of having a short alphabet, is you need to have long words. So 20 amino acids looked an awful lot like the English alphabet. Think about the stuff you could spell out if the information that's passed from one generation to the next was information that was stored in the specific sequence of amino acids and proteins. We're on to something, right? Maybe that looks pretty good. Certainly much more promising than spelling words out with just 4 nucleotides in DNA. So a lot of people were thinking protein in the early 1900s. Protein, that's where heredity is based. Griffith comes along with his dead mice with the little black X's on their eyeballs and says, wait a minute here, protein might not be such a good candidate because protein can get damaged by heat, DNA can't. So what happens next? A glimmer, the DNA is the thing that's involved. The thing that happens next is some folks build on Griffith's insights, and the next folks that really show up in this, well, first there's Avery, Griffith, and McLeod in '44. They were able to break apart the Streptococcus pneumoniae and isolate just DNA from smooth bacteria. So they're not alive, it's just DNA. And they mixed that DNA with live rough bacteria and showed that they could do the transformation. You would have thought that that would have kind of nailed it down. There's no proteins in the mix. So an incremental step, it took about 20 years for Avery, Griffith, and McLeod to do that experiment and move things along a little bit further. And then there's some folks that come along in light of those insights. Alfred Hershey and Martha Chase in 1952 do some experiments with this bacteriophage. Phage is a fancy talk for virus, right? So a bacterial virus, a specific one called T2, right? T, like Terminator 2, right? So T2 bacteriophage is the virus that they were studying. It's a virus that infects bacteria, particularly good at infecting E. coli, the lab rat of microbiologists all over the world back in the 50s to this day, right? And what Hershey and Chase did with these viruses was pretty straightforward and pretty much nailed down that DNA is what it was that was storing genetic information. What they did is they played with some radioisotopes. Remember those from like the second or third lecture in the class? This is, so they used 2 particular radioisotopes. They used P32 and S35. So a radioactive isotope of phosphorus, P32, and a radioactive isotope of sulfur, S35. Okay? In every other respect, phosphorus and sulfur differs in the atomic number, not the atomic number, the atomic mass, has an extra neutron or 2, and therefore undergoes radioactive decay. Right? So you can see where these molecules go if you follow them with a Geiger counter or a piece of X-ray film. Right? So they used P32 and S35. And they grew up their T2 bacteriophage in such a way that they were just swimming with P32 and S35. That ended up being useful because T2 is a pretty simple virus. It's got only two kinds of molecules that make a T2 bacteriophage, one of these viruses. It's got a protein coat, okay, with these fancy lunar lander module legs on it to help it grab onto infecting to the E. coli that it's about to infect. So it's got a protein outer coat, and it's got some nucleic acid, some DNA. Which one do you think got labeled or glowed because of the P32? The protein or the DNA? DNA. You know why? That's phosphodiester backbone. Phosphodiester, that's phosphorus, right? So that's where the P32 is. It's an intrinsic part of the DNA's backbone, an intrinsic part of nucleic acids. And then I don't think I need to ask, right? What's going to be labeled with the S35? It's the protein. And you know how the protein gets labeled with the S35? You know. It's because of the methionine. In the cytosines, there's 2 amino acids that have sulfur as part of their structural formula. Those cause protein to light up with S35. So check out what they do. They take these viruses that are labeled with DNA that has P32 in it and labeled with protein that has S35 in it. They infect bacteria with these radioactive viruses, and they check after about 15 to 30 minutes to see where the radioactivity is in the infected cells. Right? So cells never seen these viruses before. They get exposed to the virus. 15 to 30 minutes after the viruses have had a chance to infect them, they stop everything and they simply look to see where's the radioactivity. Guess what? They find the S35 is all outside the cells. Okay? No S35 gets inside the cell in 15 to 30 minutes. And they find that the P32 is inside the cells that have been infected. All right? So the first thing that happens in infection when T2 infects an E. coli is it squirts its DNA into the cell and the protein stays around on the outside. Those cells that just had P32 in them had no problems making more T2 bacteriophage. All right? Pretty straightforward conclusion. They were able to follow the molecule into the cell with these radioactive isotopes, and they say pretty definitively, despite all the people who are saying that there's not enough complexity in DNA, that it has to be protein or some other molecule, they say pretty definitively DNA is the genetic material. That's in 1952. And that sets the stage then for Watson and Crick to come along. And so here's their actual experiment. You want to see the pretty pictures? This is out of your textbook. It's fun, right? So they have some bacteria here that are labeled just with the radioactive sulfur, some that are labeled with just the radioactive phosphorus. They did it sometimes where both were going on, but also some when it was just one. And check out what happens. They used blenders to stop the process, right? You guys think blenders are some modern invention? They had blenders back in the 50s, and they put them to good use in microbiology labs. But they would use blenders to knock the viruses off of the bacteria, and then they'd look to see where the radioactivity was. They'd put the slurry into a test tube, spin it in a microfuge or centrifuge. The cells would sit at the bottom. The media, the stuff that's outside the cell, would float around on the top. And what they found is the radioactive sulfur at the beginning of the infection was always on the outside, and the radioactive DNA, the phosphorus, was always on the inside. It was in the pellet. That's where the radioactivity was. Here, it's in what's called the supernate. So that's the Hershey and Chase experiment. And again, that sets up the stage for Watson and Crick to come along, because that work gets done, this viral inheritance stuff, that gets done in 1952. So again, it's setting up the stage for Watson and Crick. At about the same time, another guy named Chargaff, who was a chemist by training, was getting the idea, hey, maybe this DNA molecule is important, All right? So he starts to characterize it a little bit. He found, who knows what made him think that this would be an interesting thing to do. He got 4 different species of sea urchins, all right? And he cracked open the sea urchin cells, purified their DNA, and was able to determine, he quantified how much A, G, C, and T there was, all right? Who knows? He's a chemist. Chemists do strange things. You guys have met some chemists at Wright State now, right? So yeah, come on, seriously, they're a little bit out there. So this guy, he's cracking up. You're not laughing, right? It's true. There's humor in the truth of this. He's cracking open these sea urchins, 4 different species of sea urchins, and he finds something that's kind of curious. Okay? He finds that if all of the DNA has to add up. G, A, T, and C has to add up to all the DNA. There's nothing else. But he found that one of the sea urchin species he was playing with always had a particular ratio of G to A to T to C. He found that it was about 25% G, 25% A, 25% T, and 25% C. So every time he would isolate DNA and look at the relative abundance of G, A, T, and C in this one sea urchin species, it was always the same. Okay? In another sea urchin species, he found that he was getting 30% G, 20% A, 20% T, and 30% C. Adds up to 100, but always the same and always different than it was in that other species. In the third species, he gets 20% G, 30% A, 30% T, and 20% C. Adds up to 100, right? No biggie there, but always the same and always different than the other 2. And on the third one, or the fourth one, he gets a different ratio still from the other 3. And based on those 4 different species of sea urchin, Chargaff proposes what is now known, and quickly became known, as Chargaff's rule. That's why it's up there on the projector for you right now. Chargaff's rule, you could figure it out from what I've just told you if you sat and you thought about it just a bit. But Chargaff's rule boils down to this. For every G, there's a C in DNA, right? If you have an organism that has lots of G in its DNA, it also will have lots of C. And for every A, there's a T, right? That's Chargaff's rule. And vice versa, for every T, there's an A, for every C, there's a G, right? And it works out interestingly because what he found he could do is he didn't need to do all the work of figuring out how much G and A and T and C there was. All he needed to do was figure out how much G there was, right? Because if you tell me that an organism is 35% G in terms of its DNA, I know it's also 35% C, because for every G there's a C. And 35 plus 35 is something, what, 70-ish, right? You guys are snoozing today. What's going on? That was funny. So you get 70, right, for the G plus the C. That leaves 30 behind. How do you divvy up the 30 between the A's and the T's? Let's just go right down the middle. 15% A, 15% T. He only had to figure out what fraction of the DNA was any one of those nucleotides, and from that he could tell what the fractions of all the others would be. Right? That's Chargaff's rule. So, picture yourself, late 1952, fall of 52, Hershey and Chase, DNA is where the action is. That's where heredity is rooted. That's where it's based. Chargaff has these goofy rules about DNA. For every G, there's a C. For every A, there's a T. Yeah, whatever. Good figuring that out, Chargaff. Thanks for your help with that. And lots of people in the biology field knew that if they could figure out how it is that DNA stored genetic information, that they were going to get a Nobel Prize. Nobel Prizes are a big deal today. They were as big a deal back in the early 1950s. You get a Nobel Prize. You know what happens if you personally get a Nobel Prize? You know what happens? Well, you get lots of money, there's lots of fame. You are guaranteed a cushy job for the rest of your life, right? There's not a university in the world that wouldn't hire you, pay you more than anybody else on the faculty. They're probably more than the president, right? You are set. Lots of people wanted that Nobel Prize. Among them, Linus Pauling. Okay? Have I talked with you guys about Linus Pauling, the nature of the chemical bond? I don't think I have. Here, so a little bit more about me. This class is all about me, ultimately, right? So, a little bit. Who's the person that you're most proud that you've had a chance to actually meet? Right? For me, it's Linus Pauling. I had dinner with Linus Pauling. He and I had a conversation. None of the rest of you can do that. He's dead, right? So I got you on that one. You can't beat me there. I had a meal with Linus Pauling. I think that's really cool because Linus Pauling is the only person to have gotten 2 Nobel Prizes in different disciplines. He got a Nobel Prize for peace because of the anti-nuclear weapon stuff that he did, and he got a Nobel Prize for figuring out the nature of chemical bonds, all that valence stuff that we talked about a month and a half ago. That's all Linus Pauling. So he got 2 Nobel Prizes, and he knew, well, he had already got one under his belt, and he knew it was pretty cool, and he was thinking, he's going to get himself a second one for figuring out how it is DNA could be the heritable material, right? He didn't. He didn't win the race. You know, Watson and Crick figured it out before Linus Pauling did. He did get another one for his anti-nuclear proliferation work that he did subsequently. So he got his 2. He could have had 3. That probably would have been a completely unassailable record. So Pauling is interested, and he makes it well known that he and his research group are going to try to figure out what it is that lets DNA be the genetic material. And there were others. And among the others were this pair of upstart graduate student types, postdocs, recently minted PhDs, dime a dozen, right, not that big a deal, at Oxford and Cambridge, Watson and Crick. And they think, boy, a Nobel Prize would be really nice, right? That would set us up for the rest of our careers. Why don't we see if we can't figure it out? And so despite the fact that a lot of people, prominent people like Pauling, were actively trying to figure out what it is that let DNA be the genetic material, Watson and Crick go after it as well. And so Watson and Crick start doing this work, and they pull together all the information that they can to help them figure this stuff out. They, like Pauling, like so many others, like you, knew something fundamentally useful and important, and that is that in biology, structure and function are intimately related. And so the key to figuring out how it is that DNA was the genetic material, everybody appreciated the answer to that puzzle would be figuring out the structure of DNA, right? So you guys appreciate structure and function are related. They appreciated structure and function are related. They figured if we could figure out how DNA molecules get put together, what their structure is like, we will then, it'll be obvious how it is that DNA can be the genetic material, right? So they go out and they try to figure out what's the structure of DNA. And think about the information that they had at their disposal. They knew DNA was the genetic material from Hershey and Chase and Griffith and Avery and McLeod, right? So they knew that, and that helps them. They've got Chargaff's rule. Wow. You know, for every G there's a C, for every A there's a T. At this, you know, when you're starting out, that doesn't help too much, right? But they got Chargaff's rule that they knew they'd have to figure in to whatever structure they came up with. And they had just a couple other glimmers of information. They knew the structure of guanine. They knew the structure of adenine, thymine, and cytosine, right? Those had been chemically worked out. They knew what those structures were. They knew the rough dimensions, you know, a nanometer across, literally, for a purine, and like 2/3 of a nanometer for a pyrimidine, right? So they knew those dimensions. And they had one other thing. Watson it turns out, has a photographic memory, and he was shoving around being friendly to a woman whose lab was just down the hall, Rosalind Franklin, who was a particularly good X-ray crystallographer. She was making X-ray crystal structures of things and using the diffraction patterns. When you shine an X-ray at these crystals of molecules, they glimmer in a particular way, and from that she was able to infer the position of atoms and some dimensions. Watson took advantage, by most accounts, of his relationship with lonely Rosalind Franklin, who nobody else in the building would seem comfortable talking with, and he got from her some little tidbits of information because she had been doing some X-ray crystal work on DNA, and she knew that there was an important repeating feature in DNA that was 1 nanometer, and another was 2.3 nanometers, and another that was 3.4 nanometers. So she had these 3 recurring features. Something about DNA happened again and again and again, every nanometer, every 2.3 nanometers, and every 3.4 nanometers. So he had these little bits of information. Chargaff's rule, the structures of nucleotides, and these 3 numbers that Rosalind Franklin had come up with from her X-ray crystal structures. And you know what they did? They literally went out, you know, the modern day equivalent would be Legos, but you've seen them in chemistry classes and labs, right? The ball and stick, literally balls of wood with wooden sticks that connect them. They went out, they got a bunch of ball and sticks, essentially Legos, Lincoln Logs perhaps, and they built up guanine, and they built up cytosine, and they built up adenine, and they built up thymine, and they tried to smush them together in some way, stack them, smush them, you know, all different possible conformations of these wooden models to see if they could come up with a way that they would get those recurring periodic features, the 1 nanometer, the 2.3, and the 3.4 that Rosalind Franklin thought was going to be important. And that's all they did. For a couple of weeks, they sat around in their free time and they were playing with these wooden models, trying to put them together in some way with no idea of how it is that the structure was going to give rise to a heredity, but thinking that once they got the model built up, the model was going to tell them how heredity worked. And they struggled for weeks. And one of the main reasons they were struggling is because for some reason they had it in their head that it had to be a triple helix or a triple-stranded molecule. They thought there would be 3 nucleotides coming together as opposed to 2. You guys know already how this works out. And it was just one day, it's like, they knocked it off the table, and they end up there looking, they say, what are these 2 sticking together? Hey, wait a minute here. When you have a guanine next to a cytosine, that gives you one of the 2 numbers, one of the 3 numbers we're looking for. And the thickness is another one of the numbers. And what if we stacked them in a way that sort of like spiraled up? Oh my gosh, that's the other number, that's the 3rd number. They came up with this structure. Oh, this is Chargaff's rule, by the way. So those numbers are consistent with Chargaff's rule. Okay, so here's one of the sea urchins. Look at that. This number for adenine is the same as thymine. This number for guanine is the same for cytosine, for all intents and purposes. All right? But Watson and Crick take those little glimmers of information and they figure out that if they have a phosphate group attached to a sugar, attached to one of these nitrogenous bases in this particular way, that they stack up in a way that conforms to some of these predictions that Rosalind Franklin had. I think I got a slide that shows you it. So here's some of Rosalind Franklin's X-ray diffraction photographs. This is where the crystal would have been, and then X-rays would have been shot at it from either this direction or this direction, and a piece of X-ray film nearby would get where the light got. I tell you, it's a bunch of voodoo, right? X-ray crystal structures. There are people who can look at that and they can say, oh, look, there's a carbon next to a nitrogen. Right? I mean, it's just amazing stuff what X-ray crystallographers can do. But notice these dots. There's a dot there, 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 and there. Those are the 3 periodic repeating features with the DNA molecules that she saw. And that told her something about 1 nanometer, 2.3, and 3.4 nanometers. And Watson and Crick, or I'm sorry, 0.34 nanometers. And Watson and Crick take her insight, take Chargaff's rule, and they stack their model up in this way. The 1 nanometer is essentially the distance between the beginning of a nitrogenous base on one side of the strand and the end of the nitrogenous base on the other side of the strand. So a purine nitrogenous base paired up with a pyrimidine nitrogenous base from wrist to wrist, from nitrogenous base to nitrogenous base, purine to pyrimidine, is 1 nanometer. So they had a pretty good feel for that one right out of the gate. G's base paired with C's, or hooked up with C's. So the 1 nanometer wasn't too hard for them to figure out. The 0.34 nanometers wasn't too hard for them to figure out. That's the distance that would have, if you stacked one pair of nucleotides on top of another, you would get this 0.34 nanometer periodicity. The 3.4 was tough. That was a tough one. And what it turns out to be, that's the structure of DNA, right? You don't need me to tell you that. But what it turns out to be is 3.4 nanometers is the distance from one turn of the staircase to the next turn on the staircase. As you're going up this spiral staircase, from one turn to the next was 3.4 nanometers. So when they put that twist in to the DNA molecule, they were able to do it in such a way that it gave them that 3.4 nanometer periodicity. All right? So they have these 3 bits of information from Rosalind Franklin. They have Chargaff's rule, and they have these models that only fit together when you have a G with a C and an A with a T. And that, in the span of about 5 weeks, from start to finish, they come up with the structure of DNA. And they knew this was a big deal. They knew they beat Pauling to figuring this out. They quickly wrote a paper up to publish this in a scientific journal. It's a really short paper. It's one you might even like to read. It's 1 page long. The date and page numbers are in your lecture notes if you want to look it up. It's a 1-page long paper. It's a famous paper. It's short because they didn't want to take a lot of time writing up a fancy convoluted paper. They wanted to lay claim to the fact that they figured out this structure first, and they wanted to get the word out real fast. So it's a short paper, and it describes this structure. It's got a picture of this structure in it. You know, when I was in graduate school, I had to take a class where it was called Classic Papers in Molecular Biology, graduate level class. And you know what they had us do? You know, it says something about the way that PhDs get trained. For each of these classic papers, and I think there were like 10 of them, 1 a week, something like that. For each of the classic papers, our job as graduate students would be to read the paper, study it really carefully, learn everything we could about it, and you only got an A in the class if in the course of your studying it and evaluating it, you found at least 5 errors. Right? That's how you got an A in this class. And so I can tell you there are 6 fundamental errors in the paper that Watson and Crick wrote in 1953 that describes the structure. They confuse purines and pyrimidines at one point, right? So it's simple freshman biology stuff that you guys know they're getting wrong. But there's 6 things, and if you read it, I think you'll have no trouble finding 3 or 4 of these mistakes. But it speaks to the fact that they rushed it out the door as fast as they could so that they could be the ones who got credit for figuring out the structure of DNA. You know what messed Linus Pauling up? Is he never got off of the idea of the triple helix. Right? He was just kept beating his head against, how do I get 3 strands to work? For some reason, he got it in his head it was 3 strands. Watson and Crick suffered from that for a little while too, but they moved past it. Pauling never got away from that until this structure was made public, and you're like, oh yeah, okay, this is it. So let me point out to you some of the features that make this structure such an obvious answer to the question, what's the genetic material? All right? Let me point out a couple of things to you that are going on here. It's double-stranded, right? You see there are 2 strands. Here's a strand, a phosphodiester backbone with its nitrogenous bases attached to the sugars. Here's a strand. strand 1, strand 2, right? So it's a double-stranded molecule. It's going to give us a double helix. Each strand, 1, 2, double, right? Notice that it's got this phosphodiester backbone here, and it's got this phosphodiester backbone here, right? So that's the same on both strands. But notice this. The strands, the word for it, the strands are anti-parallel, right? Everybody knows what parallel means. This strand is parallel to that strand, right? But they're anti-parallel because this strand, to a molecular biologist, this strand starts here and ends here. There's a 5' end and a 3' end. And on the other strand, it's the opposite orientation. It's anti-parallel. It begins here and it ends there. The reason I say it begins here is that when you add new nucleotides onto a DNA molecule, you always do it at the 3' end. You never tack more nucleotides onto the 5' end of any nucleic acid, RNA or DNA. They only get added on to the 3' end. So you start here and you end here. And you know what? You know why they call it the 5' end? I should probably be explicit about that, shouldn't I? It's the 5' end because let's count the carbons in this sugar. That's carbon 1. That's the 1' carbon going clockwise around the circle. So carbon 1, 2, 3, 4, and there's 5. The phosphate group is dangling unattached to anything here off of the 5' carbon. That's why it's the 5' end. Down here, you've got a 1, 2, 3. You've got a hydroxyl group that's dangling unattached to anything off of the 3' carbon. Right, so that's the difference between 5' and 3'. Cells treat the 5' end of a nucleic acid as the beginning and the 3' end as the end, and you only add nucleotides onto the 3' end. So DNA synthesis and RNA synthesis always goes from 5' to 3'. I'm going to say that to you a bunch in the course of the next couple of lectures, but you always go 5' to 3'. For a DNA, 5' and 3', it's equivalent to and it's every bit as important as left and right, right? If you're trying to figure out the information stored on a page of written lecture notes, you really got to know. It helps a lot, right, if you know to read from left to right, right? Who knew? It's not very helpful to go from right to left. It only makes sense when you go left to right. Same with DNA. Reading in the other direction, just gobbledygook. Reading left to right, that's where the information turns out to be. So that's one of the things that Watson and Crick find out, is this only works if they're anti-parallel. That was an important insight that they had. And the basis, the molecular basis for Chargaff's rule, for every A there's a T, is base pairing. The structure of thymine is complementary, that's the word, to the structure of adenine. There's a capacity to form 2 hydrogen bonds between every thymine and every adenine. You guys remember hydrogen bonds, right? There's a little bit of a negative charge on one side and a little bit of a positive charge on the other, partial separation of charge, and they offset each other a little bit. 2 for every T and A that pair with each other. Nitrogenous base, base pair. That's where that language comes from. So there's a pairing of those bases. They literally stick together because of the stickiness of these partial charges at the T and the A. So T's and A's can form 2 hydrogen bonds. G's and C's can form 3 hydrogen bonds when they come and they get butted up against each other. So it takes more energy to peel apart a G and a C than it does an A or a T. One more extra hydrogen bond. If you try to base pair an A with a G, and Watson and Crick tried this for quite a while. If you tried to base pair an A with a G, you find you got 2 big problems. One of the problems is that the two purines are too big, right? You kind of bulge out on the double helix. It's not going to be 1 nanometer anymore. It's going to be like 1.5 nanometers. So there's too much stuff going on here if you moved this down to here. And the other problem is that G's and A's don't base pair at all. In fact, they're the opposite of base pairing. They're repulsing each other. Their positive charges are butting up against other positive charges, and their negative charges are butting up against negative charges. So G and A can't base pair. G and T can't base pair. C and T can't base pair. C and A can't base pair. These hydrogen bonds, the spacing is wrong, the charges are wrong. Only T can hook up with A, and only G can hook up with C. Okay? So these are things that Watson and Crick are discovering with the structure that fits these three important dimensions that Rosalind Franklin had given to them. And when they get this structure, the expectation is the structure then says it all, right? Say no more, right? There you go. Here's the structure. Do you need me to explain to you how it is that this is the genetic material? Do you? I don't know. You probably have got. You can look at that structure and figure it out probably just as well as I can explain it to you. The information is stored in the linear sequence in which the nucleotides are arranged. Okay, fine. 4 is a pathetically small alphabet, right? But the price you pay for having a pathetically small alphabet is you just have long words, right? So, okay, we can live with that, right? So the information is stored in the order in which the nucleotides are arranged. And there's a redundancy in the information here. What you see on one strand, you can use to predict with certainty what you'll find on the other strand. And that then turns out to be really helpful, because when you're dividing a nucleus, when you're replicating a cell, the splitting of the two strands apart and using each of those two strands as a template to make another pair now of double-stranded molecules, that's sort of the, oh yeah, of course, that must be how it works. Why were we thinking triple helix? What's up with that? Double, because when you divide cells, you divide in two, and what you do is you split the strands in two. The information in one strand and the information in the other strand is all you need to have locally to be able to make a new complementary strand and now have two double-stranded molecules where once you had one. Okay? So the structure lends itself to that. And I'll tell you what, if you do look up that paper, pay particular attention to the very last sentence in that one-page long Watson and Crick paper. I think I know it by heart. I may not get it exactly right, but it's essentially this. The structure that we propose for DNA suggests a possible mechanism by which the genetic material may be replicated or copied, right? So they say that. And they're waving at this idea that you separate the two strands and you do it. They didn't spell it out. They didn't spell out what I just told you guys. You separate the two strands. The information on one strand is enough to give rise to a double-stranded molecule, and it makes sense. It all works. They didn't spell it. You know why they didn't spell it out? It's beautiful why they didn't spell it out. Again, these are people, right? These are guys. Maybe that's part of the problem. They're kind of nerdy guys on top of it. Maybe that's also part of the problem. You know why they didn't spell it out? Because they knew they were going to get a Nobel Prize for that structure, and they didn't want to mess it up and compromise, get any tarnish on their Nobel Prize by saying something in this paper that was wrong. They still had 6 errors, but they didn't want to say this is how DNA replicates and then have somebody else come along later on and say, you morons, that's not how DNA, that's stupid, that can't be how it is. So they just wanted to do this. They wanted to say the structure suggests how DNA might replicate. They didn't want to pin themselves down on exactly how it is that DNA might replicate, but they confessed to this in their autobiographies, but they wanted to make sure that if somebody else came along and said, this is how DNA replicates, that they'd be able to say, of course that's how DNA replicates. That's what we meant all along when we put that sentence in at the end of our paper, right? Is that slimy or what, right? They didn't want to put they wanted all the reward, but they didn't want any of the potential downside of coming up with a stupid idea, right? So that's, again, it's a beautiful paper. It's interesting to see, especially when you know some of those little nuances of what's going on. But it turns out the structure really does tell you how it is that this stuff gets replicated. And there it is. It's a ball and stick three-dimensional space-filling model of what DNA looks like. And I've already talked with you about all this stuff. If you try to hook up two purines, it's too wide. You try to hook up two pyrimidines, it's not wide enough. A purine and a pyrimidine, just right. It's the Goldilocks sort of combination. Here's the base pairing that I was talking about between an A and a T. There's two hydrogen bonds. Check it out. A little bit of negative charge there, a little bit of positive charge there. Nature likes this. Check it out. A little bit of positive charge here, a little bit of negative charge there. Nature likes it. Check it out. Negative, positive, base pairing, hydrogen bonds. Okay? Got the double-stranded helix, two-stranded helix. We got base pairing. There's complementary strands. And let me say a few words to you here now about the rest of this outline, and we'll talk a bit about DNA replication. I think I've mentioned to you guys, it's not a secret, before I was at Wright State, the job I had before I came to Wright State is I was a postdoctoral researcher. It's the formal title. Everybody says postdoc, but I was a postdoc at Harvard. Okay, and being at Harvard was cool. It was a nice place to hang out. I wasn't there for very long. It was about a year. And I lived my postdoc life to the max. I took full advantage of everything that Harvard had offered. Did you know that postdocs at Harvard are considered faculty? How about that? And faculty at Harvard get to go to the faculty club, right? They have afternoon high tea at 4 o'clock every day. I was there, you know, give me my free tea from the Harvard faculty. I've had tea with John Kenneth Galbraith, you know, a Nobel Prize winning economist. I've had tea with Wally Gilbert, a Nobel Prize winning molecular biologist. I've had tea with Wally Gilbert and John Kenneth Galbraith at the same time. Boy, the conversations in the faculty club are really cool. But among the other things that you get to do is, as a faculty member, postdoc, I could go sit in on any class that I liked, right? And so I charted it out. Stephen Jay Gould was giving a lecture about the Burgess Shale in his class. You guys may not know what that is, but whoa, Stephen Jay Gould talking about the Burgess Shale. I'm there. That's what he's famous for. So I'd go to his lecture on that. And it happened that Matt Messelson was on the faculty at Harvard at the time. And I asked somebody, I said, is that the Matt Messelson of the Messelson-Stahl experiment? And he's like, yeah, what's he teaching? He said, chemistry, something about DNA replication. I'm going to go sit. I've heard Matt Messelson give a lecture on what I'm about to tell you about here. Matt Messelson and his friend Frank Stahl saw Watson and Crick's 1953 paper that describes how DNA's structure lends itself to being the genetic material. And Messelson says to his friend Stahl, hey, Frank, you know, I think they're onto something. I think that is how it is that DNA could be replicating. And you know what? Why don't we put that to the test? Let's test this hypothesis, right? And Stahl says, yeah, sure, Matt, let's do it. We got the radioisotopes. We can do this experiment. Let's play it out and see how this works out. And so Matt Messelson and Frank Stahl do what has become known in '54. They do what's become known as the Messelson-Stahl experiment. And what the Messelson-Stahl experiment is, so the beauty of hearing Matt Messelson give this lecture, and incidentally, I got that on you guys too. He's dead now also, so can't beat me on this one. Matt Messelson, when he gets to this point in the lecture, he says, so, folks, you know, the structure suggests how it is that DNA replicates, so this is what we did. It's so cool. Matt Messelson says, so this is what we did. This is what Matt Messelson and Frank Stahl did. They have this picture in their mind of a DNA molecule, right? And it's dark blue because in their minds, they had it made with heavy nitrogen, an isotope of nitrogen that's heavier than the normal nitrogen that we usually have. Nitrogen 14 is the normal nitrogen, nitrogen 15 is what they had their hands on. So they grew up some bacteria, with nitrogen 15. Why do you suppose nitrogen's useful here? nitrogenous base. The nucleotides have lots of nitrogen in them. So they grow up their bacteria with heavy nitrogen, dark blue, right? All the DNA is denser, it weighs more because it's been growing in nitrogen 15. And then, when you replicate the DNA, you separate the two strands, right? And what if, at this point, you fed the bacteria no more of that nitrogen 15 for you? Now you give them nitrogen 15. You have them start making their nucleotides out of a different kind of nitrogen. What should happen is that the DNA that you synthesize in this next round of DNA replication should have an intermediate density, right? One strand should be made with the heavy nitrogen, the other strand with the light nitrogen. They're an average density. It's not heavy anymore. It's not light. It's somewhere in between heavy and light. One strand's heavy, one strand's light. Matt Messelson and Frank Stahl predict that if we grow bacteria with heavy nitrogen, they'll have heavy DNA. If we let them grow one generation with light nitrogen, they'll have intermediate density or weight DNA. And if we grow them on light nitrogen, it's just going to be light DNA. So they predict this outcome, and that is essentially the Messelson-Stahl experiment. It proves Watson and Crick's idea that if you separate the two strands, you use one strand as a template, and you lay down a new strand opposite that template strand. This is as an alternative to the idea that, so they had, they predicted other alternatives. I think I have pictures. Let me see. Yeah, so this is the Messelson-Stahl experiment. So what they were suggesting is that it would be a semi-conservative approach. You start with blue, dark blue, dark blue. The first generation you have a dark blue with a light blue. The next generation, of the next round of DNA replication, you'd get some of the DNA that would, half of the DNA would be intermediate density and half of it would be light. All right? That's what they called semi-conservative. It's conservative in that one strand, that the original strands don't change, but it's only semi-conservative because the new strand is completely new. That is an alternative to a conservative model, where the two heavy strands stay associated with each other, and your new molecules are completely new, made with lighter nitrogen. So that would be a conservative model, and an alternative would be a dispersive model. Take a look at the outcomes after the second round and even after the first round. If you separated out the DNA molecules on the basis of their density, the semi-conservative model after the second round of DNA amplification would have one band of density of DNA that was in the middle and one band that was floating at the top because it was less dense. The conservative model would have had one band, so I'm using my hands here. They would, the semi-conservative models, a band's here and a band's here, right? Equal intensity. The conservative model has a band here and 3 times as big of a band here, right? This is different than this, visibly different than this. And for the dispersive model, what they would have seen would have been just sort of a smear, right? Somewhere in the middle. Not a band and a band, but a smear, right? Smear is different than this and this, and this is different than this and lots of this, all right? So Messelson and Stahl do this experiment, and they look to see how the bands change from the first round of replication and then in the second round of replication, and they come away and they say, Watson and Crick were totally right with what they said was how DNA replicates. And I talked with Matt Messelson afterwards, and I said to him, how did it make you feel when you read Watson's autobiography? And he said that the word that he used is he pissed on the idea that they knew how DNA replicated. He said, how did that make you feel? It's like, oh, well, you know, you got his Nobel Prize. It's just what happens. Nobel laureates do stuff like that. Right? Matt Messelson was a pretty cool guy. So that's the Messelson-Stahl experiment. And they're even better pictures than my hands. I didn't need to do the hand stuff today. And even more times I didn't need to use my hands. So you guys, if you couldn't see what I was doing with my hands, look at the PowerPoint that's available on Pilot, and you'll be in good shape. All right. Well, in the couple minutes that we have left, you've got a pretty good sense, I think, now of what I mean by semi-conservative. I'll tell you that DNA begins to replicate at discrete spots along the length of chromosomes. Those spots are cleverly named origins of replication. The enzyme that does DNA replication is called a DNA polymerase. I won't be able to do DNA polymerases justice in the last minute or two of class, so I'll probably say a little bit more to you about them in lecture on Tuesday of next week. But I'll tell you this, and I've already suggested this to you, they are insanely fast. Right? Remember when I talked with you about how long it takes to replicate E. coli's 3 million nucleotide long chromosome? 2,500 nucleotides a second fast. Right? Think about how fast 2,500 nucleotides a second is. And bizarrely accurate. Less than one mistake per 30 million nucleotides that get incorporated. Right? So crazy fast and crazy accurate. And I'll tell you a little bit more about them later. And there's some other proteins that are involved. So this is a replication fork. I'll try to start with this at the beginning of lecture on Tuesday. All right. What is all that stuff there for? That's what we really want you to see. So there's a question I'll throw at you on the final exam for the class. I'm essentially asking you, what's up with Chargaff's rule? I'll get out of your way. So have a good weekend. See you guys on Tuesday.